everyone. Good morning or good afternoon, depending on where you are in the world. Thank you very much for joining us to this 7th webinar offered by the Groupe Polyphenols. The Groupe Polyphenols Society is an international association founded in France in 1972 with the aim of promoting research on plant polyphenols from their most basic and fundamental physico-chemical properties to their most diverse applications in food and agricultural, pharmaceutical, and cosmetic sciences and technologies. The Groupe Polyphenols started to develop this series of webinars in polyphenol research in 2022 under the initiative of President Denis Baron, with presentations about polyphenols in soil systems, food and beverages, and about their characterization. If you didn't have the chance to participate in the previous webinars, you can have access to the recordings on the website of the Groupe Polyphenols. I'm Dr. Aude Watrelot, an assistant professor and extension specialist of enology at Iowa State University, focusing on polyphenols' chemical properties in wines. I'm the vice president of the Groupe Polyphenols, and I'm co-hosting the webinar today with Dr. Stéphane Quideau. Stéphane?
Hello.
So before I give the floor to Dr. Quideau, I would like to provide you some instruction. So the webinar is currently being recorded and will be available on YouTube in about a week or so. Please type your questions in the chat box that you can find at the bottom of your screen by a click on the 3 dots. Your questions will be answered at the end of the webinar. At the end of the webinar, you will also receive an email to complete a Qualtrics survey about how we did today and what we will need to change for future webinars. So please take a few minutes to complete the survey. That's really important for us. Also, if you need a certificate of participation to this webinar, please send me an email to request it. You will then receive it within a month. So now it's the time to start our webinar. Dr. Quideau, the floor is yours to introduce yourself and our speakers today.
Thank you very much, Aude. So good morning, afternoon, evening, everybody, depending where you are. So I am Stéphane Quideau, professor of organic chemistry at the University of Bordeaux in France, the southwest part of France. I'm a former president of the Groupe Polyphenol, and my group for many years has been working on different aspects of polyphenol, studying structure, chemical reactivity, total synthesis of some compounds, and chemical biology aspect of bioactive plant polyphenol. But let's say a few years ago, I did, during my PhD, I have the chance to study lignin chemistry in John Ralph's laboratory at the University of Wisconsin in Madison. So talking about lignin today and hosting this webinar is really a special honor. But before digging into the chemistry of lignin in plant cells, I would like to give the floor to Dr. Juha-Pekka Salminen, who will introduce the upcoming international conference on polyphenol that will take place in Turku next July. JP?
Right, thanks, Stefan. Let me share my screen. Okay, hopefully you can now see the website of the conference, ICP 2025 turku.youtube.fi, and hopefully many of you were already planning to come to Turku in 2020. You know, that was our intention then to organize this conference live, but unfortunately pandemic hit the world at that time, and 21 we organized this conference only remotely live. So now you have the chance to come to Turku for real. And let me go through the website a little bit. So you can now see that we have opened the abstract submission for you. So if you are willing to come to Turku on 7th to 10th of July 2025, when of course Turku and Finland is at its very best, you would now like to submit your abstract. And on the basis of that, you will be then maybe one of the lucky 147 oral presenters in the 21 sessions we organize at the ICP. So now please go to the page where to submit your abstract, and you will be then evaluated for the poster or oral presentation. And while going through these pages, you will also see that we offer you also many different types of options in Finland, just for your holidays, for example. But let's go through the scientific program if you're interested how that it looks. So you can see the preliminary schedule over here. And when you click it, it will be larger, and you will actually see that we will have several sessions also for the young students, especially. So we try to encourage PhD students and postdocs to have presentations as well. The topics are the very, very typical ones. We have little bit updated them, but you know, the structure, reactivity, synthesis, bioactivity, metabolomics, food processing, biodiversity, biomaterials, etc. So we practically can cover all of the aspects of polyphenol chemistry quite efficiently. So you will certainly find your own topic in here where you would like to have your oral presentation. And if you look at the invited speakers, you find familiar faces like Stéphane over here. You will see many other good polyphenol scientists like Rosa Lamuela-Raventos, for example, Pedro Menia, you will find in here. You will see Monica Giusti and so on. So you can get familiar to the presenters, the plenary presenters over here, if you are interested of them. And when you go to the registration button over here, you will see that actually we have quite affordable registration fees at the bottom. So you can see students are pretty cheap, but also the normal prices are cheap, especially if you are Groupe Polyphenols member. So you can see that you can save up to 200 euros by becoming a member of the Groupe Polyphenols, and that's only 50 euros payment you have to pay to save 2,000 euros. And you will have the links over here. Online registration is available for you. You can do it now, or if you do it before 1 May, you will get the discount, and after 1st May it will be a little bit more expensive, but not too much. And here are links if you want to save by joining the Groupe Polyphenols. You can see the links over here, and you just follow them, and that's it. And we also offer you a unique gala dinner in the oldest castle in Finland, and that you will then have to pay separately the gala dinner price, and we will sell the tickets to you separately. Communications, that's practically about abstracts. So if you go over there, you will now see the topics again. You can choose your topic, and there are many guidelines how to do it. So you can have one submission per ICP participant, and you will simply register in Oxford Abstract System. You push this button, and then you can register. And while you have registered, you can start submitting your abstract that actually will be then published as Polyphenols Communications with a citable ISN number. You can see an example of the abstract as well, so it will then show how it looks. This is just an example I made for you, so sort of very simple to do. You can add one figure if you like, one image if you like, and follow just the instructions, and that will be very clear. If you have any questions, simply fire away on these email addresses over here. And as you know, ISSN number will be given to your abstract later on. Venue, we will be located at the very heart of the Turku city, so close to the Aura River. Actually, pretty lovely place. You will see the riverside. That's the fanciest place in Turku in the summertime, and also the Turku City Theatre where we organize. The venue is pretty modern, so everything should be pretty fine in the venue as such. And also accommodation, we have done it now, pre-booked the hotels for you. So if you can already now look at the hotels we have pre-booked for you. You can do the reservations if you like, as you can see. So you can scroll through the options as much as you want. Social program, we will have a welcome reception, of course, hosted by City of Turku, and the gala dinner, as I said, in the Turku Castle, but also then other activities if you want to go to Moving World, for example, with the children that you have with you, or you want to have other types of cultural experiences. You can just click these and book a kayaking day, for example, in the Turku archipelago, which is of course pretty nice, even the whole day for kayaking in the archipelago, and so on. All the options are available for you just clicking here. Very important information is the Groupe Polyphenols grant. So if you want to apply for the grant, while you submit your abstract. Here I advise how to do it. So Groupe Polyphenols will offer grants to postdocs and PhD students, research associates. So it's a nice opportunity not to miss. So just have a look at these and where to send the information. The email addresses can be found here and applications can be found down there. So just remember to do it while you submit your abstracts. And of course, if you want to be a sponsor, partner, exhibitor, we also have the options available for you. And we give you good visibility in many ways. And Discover Turku, we are very easy to connect. So typically you would maybe love to come to Helsinki, more connections to Helsinki, and then very easily from Helsinki to Turku, 2 hours by bus or by train. You can also fly to Turku, but options are more common to Helsinki. Very good connections. And when you want to connect your holiday with our conference, here are also some links for you how to do it, how to make yourself familiar with the options we have at the Turku area, how to become a Finn, how to speak Finnish, etc. So look at these options down there. All right, but now I think the main thing for you is to click the home button and to see that abstract submission, as I told you. Please be one of the 146 presenters and submit your abstract until the end of this month. And we hope to see you all in Turku in July. So welcome.
Before we start by the presentations, I just wanted to share one thing about the how to join the Groupe Polyphenols. So that's a new membership option that we have, is a 2 years option. And so that's something that we really want to share with you. If you are interested in joining the Groupe Polyphenols, that can help you to go also to the conference in Turku. So if you want to join as a 2-year plan option, you can email the Groupe Polyphenols at that email address that you see on your screen. You will have to mention those different informations in your email, but that's the options. All right, so without further ado, I'm leaving the floor to Dr. Quideau just to do the introduction of our first speaker.
Thank you, Aude, for this last information about the Groupe Polyphenol. So let's get to science. It's my pleasure to introduce our first speaker of the day, Dr. Leonard Blaschek, who will present a lecture entitled Not Quite Random, the Compositional Control and Physiological Importance of Specific Lignin. Dr. Blaschek completed his bachelor degree in Greifswald in Germany before moving to Sweden for his Master of Science in Uppsala. And then he did a PhD in Stockholm with Edward Pesquet. The 3 core questions he tried to answer during his PhD were how to efficiently quantify lignin in situ in planta, how plants adjust lignin composition from one cell to another, and whether lignin composition carries physiological relevance. Shifting focus, he then joined Stéphane Quideau's laboratory in Copenhagen in 2022 on an EMBO fellowship, working on the coordination between environmental stimuli, carbon status, and cell wall biosynthesis. Dr. Blaschek was the recipient of the Groupe Polyphenols Raga Ibrahim Prize at the occasion of the previous ICP meeting in Nantes, France. So, Leonard, the floor is yours.
So, thank you very much, Stéphane. I hope you see everything.
Yep.
Good. Thank you very much for the introduction, and thank you very much for the prize that you awarded me last year. It was an honor. It's also actually particularly interesting to me to present today alongside Norman Lewis, because this topic that I've spent so many years here on researching, when I started my PhD and got into the literature on sort of lignification and whether there was any biological control in it, or specifically in the last step of the polymerization, it seemed like almost the entire research field had sort of embraced this very simplistic but sort of satisfying idea that lignification or polymerization was entirely random, just controlled by thermodynamics of the activated monomers, while Norman Lewis really, to me at least, seemed like the only guy still sort of advocating for a layer of biological control that we hadn't really fully understood yet, which became very relevant in my PhD. So I'm very much looking forward to his talk later. But before that, I'll talk a bit about what we did. So lignification, of course, or actually, let's start with a reminder. This came up. I watched over the weekend the talk of the previous Raga Ibrahim awardee, Bridget McGivern, I think her name was. Great talk. You should check it out on YouTube. And she mentioned that the soil held more organic carbon than the atmosphere and all life on Earth together, which then, of course, throws up the question: well, that life on Earth, where's the carbon actually sort of distributed in that? And I think what might be surprising to many people is really that when we talk about life on Earth, at least when we talk about it in terms of carbon, it's really most of it is plants, and of that, the majority is plant cell walls. So it's a nice reminder to keep to have in the back of one's mind when one thinks about the sort of the sheer quantities of these polymers that we're talking about here. And of course, since the plant cell wall that is, this big yellow box here is mostly woody plant cell walls, something between 15 and 35% of that is lignin. Now, lignin is a complex polyphenolic, which is somewhat of a tautology, of course. I mean, all of you working on polyphenolics know that they have the habit of being very annoying and complex. It is also hydrophobic, as so many, and in the plant cell wall, as sort of this illustration here from the 80s shows, it is thought to sort of fill in the spaces between the sugar-based polymers in the cell wall. The complexity of lignin, a lot of it comes from the variety of the lignin monomers, and there is various chemical forms of these monomers, but most of them share sort of a specific backbone, which is this phenylpropanoid backbone shown here. And the variety between these phenylpropanoids largely occurs on or along two axes, which is the functional aliphatic group and the level of ring substitution. So just as an example, if we look at the most common examples of these, that's the aldehyde and the alcohol aliphatic group, which I'll abbreviate with chemical terms here later on. And then the different patterns of ring substitution, which are named with letters. So for example, we have the monomethoxylated G units and the dimethoxylated S units, which make up the bulk of these ring structures in lignin. Now, what about the process that assembles or that actually polymerizes and creates lignin in the plant cell wall itself? This process is, you can roughly split up into 4 steps. The first one being that while the polysaccharidic or cellulosic cell wall is deposited, lactases and peroxidases, which are oxidative enzymes, are also translated and deposited in that, forming cell walls where they're tightly embedded. Next, the lignin monomers are biosynthesized in the cytosol and then transported into the wall. And then lastly, in the cell wall, these lignin monomers are oxidized by the lactases and peroxidases into radicals, and these radicals then undergo radical coupling with each other or with the growing polymer to form this lignin polymer. And this is where most of that randomness that we talk about comes in, because it is considered that these lactases and peroxidases have such a broad substrate range. Excuse my dog. Such a broad substrate range that they essentially would oxidize anything that comes into the cell wall that even remotely resembles a lignin monomer. The next thing I want to highlight is that lignin is not just complex in the sense that it contains a lot of different monomers and intermonomeric linkages, but that these different monomers actually are spatially distributed rather specifically in different plant organs and plant cell types. So the most well-known example of this, generally speaking, is the fact that in angiosperms, the fibers are enriched in S units, which you see here stained in red, and the tractary elements are enriched the G units. Tractary elements, of course, being the cells that are actually responsible for the transport of sap throughout the plant, while fibers have a structural support role, so to speak. And this Moeller staining that you see here specifically stains S units, so you see where the S units are distributed. Now, we still only really understand a fraction of these different specific lignins and where they're distributed, but a couple other examples are, for example, in these same cells, that if we zoom in a little bit, we see that the compound middle lamella, where two adjacent cells sort of meet, and the cell corner at the tricellular junctions, those are specifically enriched in so-called H units. And if we go through the literature, there's other examples of, for example, an H unit enrichment in compression wood, a so-called C unit, which is specific to certain clades of plants, which form these highly specific and rather pure C lignin depositions in the seed coat specifically. The Casparian strip, which is also lignified, seems to be enriched in G alcohol units. In sort of surface tissues of plants, such as bark and rind, recently, or relatively recently, a lot of atypical flavonoids and stilbenes have been found to be incorporated in rather large proportions into the lignin polymer. In defense lignins, for example, in the Solanaceae, which are ectopically deposited in response to an attack, a high proportion of that is made out of ferroyl tyramine. And then lastly, the fact that these S units, which are dimethoxylated, making them some of the most metabolically expensive lignin monomers that the plant generally produces, have evolved, to our knowledge, at least 4 different times. Now, these specific distributions of different lignin monomers and different types of lignins, when considering that the polymerization of lignin is somewhat random, is very much at odds with what we observe about the diffusion of lignin monomers. Because of course, if we assume the polymerization of lignin to be random, the only steps in the process of lignification where biological control could come in place are the biosynthesis of monomers and the transport of these monomers. But now, and we've observed this in many other scenarios as well, but this is a nice example from Wout Boyan's lab, where you, for example, here have a poplar plant where they have specifically restored lignin biosynthesis, the lignin monomer biosynthesis in vessels. You see that fibers that are close by these vessels, like here, or close by the rays, are much more heavily lignified than fibers that are sort of far away from any vessels, giving you a very good idea that these lignin monomers, once biosynthesized and exported into the cell wall, are diffusing quite a long way. And beyond this, there's even evidence that these monomers are transported long distance through the vessel or through the xylem sap. So that then throws the question, how do plants actually control the deposition of these tightly regulated specific lignins on the sort of nanometer or sub-micrometer scale? And it also offers the question of why really do they bother? Is there physiological relevance to these specific lignins in specific places? Because again, widely assuming random lignification and the sort of easy replacement of one monomer with the other, this idea that specific monomers might have specific physiological relevance, wasn't very widespread, at least when I started with this. Before I go into the results of our work, the first big problem that faced us when we started working on this was that we realized that there wasn't really any tools around that were fully able to generate the kind of data that we needed to address these questions. So in lignin biochemistry, very generally speaking, the perhaps most classical approach to lignin analysis was to take your plant, either an organ or the whole plant or some part of it, grind it up into a powder, and then run a variety of lignin analysis methods on it. Now, these methods are very, very good. Many of them give very finely resolved, chemically finely resolved data. But the problem, of course, is that you will always just get an average of all the cell types and developmental stages and cell wall layers that your sort of sample comprises. There's no space. All the spatial information gets lost. So instead, what we wanted to focus on was in situ lignin analysis. So figuring out ways to actually be able to quantify the amount and the composition of lignin directly in the cell wall layers where we were interested in them. When we started there, this is roughly how sort of the landscape of lignin in situ quantification tools looked like. So here you have on the y-axis sort of a pseudo chemical resolution scale, starting with the just tools that are able to detect the mere presence, going to the chemical elements, the units, so like S or G units, and then monomers, so the combination of ring substitution level and functional group, and structures, so methods that are even able to determine specific linkages between specific polymers. And then here you have a spatial scale on the x-axis. And there is, as I said, many very cool tools out there, and many of them deliver fantastic data given, depending on what your question is. But for our question, really, what we needed was something in this area, something that was able to distinguish specific structures, both in terms of ring substitution and functional group, on a spatial resolution somewhere close to the diffraction limit of life, so around 200 nanometers. And really the only two things here were UV absorbance, which has in the past, in the 90s, been shown to be able to differentiate between G alcohols and G aldehydes, but isn't really widely used and needs a rather specific setup, as well as labeled monomers, which are also a great tool, but crucially, they're not quantitative for the lignins actually in the plant. They are more sort of like signposts. So the idea is that you add fluorescently labeled monomers to the plant while it's lignifying, and then you sort of get the readout of where these are incorporated. But of course, they are not actually identical to the native monomers, and their incorporation isn't necessarily quantitative. Now, by the time we were done, something had sort of changed, and the two that we contributed to mainly were, that we validated ourselves, were the Wiesner test and Raman microspectroscopy. So the Wiesner test you're probably familiar with. Is also called fluorocristanol HCl. It's been used for the last 150 years to show the presence of lignin, right? To get a pink stain when there's lignin. But in recent years, sort of it was widely understood that it would stain any aldehyde units in the lignin polymer, and that, but only if at the end of the polymer as end groups. And that, of course, would then suggest that you get rather limited specificity and also no quantitative capacity. But we were able to show with some detailed work that actually the Wiesner test in cell walls only stains G aldehydes and is a reliable proxy for the total amount of G aldehydes in the cell wall. And the other method is Raman microspectroscopy, which is a vibrational microspectroscopy technique, which gives you essentially a whole fingerprint of the chemistry in the cell wall, which can then sort of be decomposed to give you various information about the chemical composition of the lignin in the cell wall. All of this we spend a lot of time sort of validating and refining. That's all published a while ago now. You can check that out. But these methods will be very important for the rest of the talk. Bringing me to the first big question, right? The how. How do plants control lignification this tightly when the monomers, as we've seen, are so widely diffusing? So given that it couldn't really be monomer transport or monomer biosynthesis that was the origin of this tight regulation, that left the polymerization of the lignin polymer, which, as I mentioned, is catalyzed by lactases and peroxidases. Now, given sort of the recent evidence, it seemed more and more like lactases were playing the key role here. So we decided to focus on that gene family. Now, there's 17 lactases in Arabidopsis. There is good evidence for an involvement in lignification for at least 7 of them. We picked 5 and stacked those, generating one quintuple mutant that we just abbreviate with Q here. And then we also generated all the different combinations of quadruple mutants that we sort of shorthanded with Q-4, for example, being the one that is like the quintuple mutant, but with an intact lactase 4, right? And there we see, if we grow these plants, we see that the rosette growth of them is actually not affected at all, which was reassuring because it sort of let us know that there was no metabolic feedback that just messed up plant growth in general, but that it seemed like that the effect that we saw in vertical growth, which was rather dramatic, really was due to the sort of biomechanical and structural effects of missing lignin. And there you see that this quintuple mutant really is dramatically shorter than the roughly half of the wild type. And the different quadruple mutants had sort of various phenotypes in between there, with some of them, like lacase 4 by itself or lacase 17 by itself, seemed to be able to restore wild type growth almost completely, while lacase 5 and 12 didn't seem to do much. Now then, first we looked at the just crude sort of quantity of lignin on the subcellular scale. For that, we could just use lignin autofluorescence. Lignin, as so many polyphenolics, is autofluorescent, and the sort of intensity of that autofluorescence, when controlled properly, is a good proxy for lignin quantity. And if we looked at, when we looked at these cells, so these are intervesicular fibers here, we realized that in all of our quadruple mutants, except for Q-12, the cell corner, which traditionally or in wild type plants always have the highest concentration in lignin, were hypolignified. As you can see here in the quintuple, for example, you see that the cell corners are actually lighter than the surrounding compound middle lamella. And indeed, in the Q-12 and the wild type, you had this increase here in the relative autofluorescence in the cell corner, while in the quintuple, and indeed in all the other quadruple mutants that were lacking lacase 12, you had a hypolignification of specifically of the cell corner. Showing us then that if anyone sort of still had the notion that these lacase paralogs were all essentially redundant, that really was not the case, and that even if we're just talking about lignin presence, we have specific sort of delineation that are performed by specific paralogs in different cell types and different cell wall layers. Then, of course, the perhaps more interesting question was whether we actually get a lignin composition change from our different lacases. A couple of years previously, we had, this was just a week after AlphaFold 2 came out, so that was very, very exciting. So I used that to just model all the lacases that had been, all the rhodopsis lacases and other lacases that had been functionally characterized to some extent, and just did a rough analysis of their substrate binding pocket topology. And if we sort of take that topology here and look at the volume, compactness, mouth area, and the sort of how buried the catalytically active copper atoms were, we saw a rather dramatic difference in these substrate binding pockets between the different lacases. The question there then, of course, being the different pockets, of course, suggest that there might be substrate specificity, but is there really one? And this is something we were, desperate to find out. Our quintuple mutants, of course, and the quadruples gave a pretty good opportunity to look at that. And if we look here at our sections and stain them with the Wiesner and the Moyle test, both of which I mentioned. So the Moyle test, as a reminder, stains S units in red, and the Wiesner test is pink, and the intensity corresponds to the amount of G aldehydes in the cell wall. We see that in the quintuple mutant, we have a drastic reduction in lignification to the point where we have essentially no G aldehyde anymore in our fibers, both intervesicular and axillary fibers, and also the S units are almost absent. But then we see that in the Q-4, for example, so as a reminder, this only has an active lacase 4. This plant phenotypically looked almost exactly like the wild type, and also from the cell morphology and most things. But if you look at the Wiesner test here, you see that the stain in the intervesicular fibers really is rather dramatically lighter, suggesting then that lacase 4, while it was very good at compensating for the loss of most lacases in terms of total lignin deposition, it was specifically unable to efficiently incorporate coniferal aldehyde, so G aldehyde. Of course, we needed to confirm this, so this brings us now to the second method I mentioned, which is Raman microspectroscopy. And if you take a Raman spectra of a lignified cell wall, this is roughly what you get. So you have the yellow spectra, each line is an individual spectrum. The yellow spectra of the quintuple mutant and the gray spectra from the wild type. And then you have different bands that are characteristic for specific lignin substructures. So you have one band here that is specific for G alcohols, one for G aldehydes. You have one for S and one for G in general, and then you can make sort of a weighted sum out of various ones that nicely represent total lignin. And we scale all of these always to the cellulose band to get an idea sort of the lignin to cellulose ratio in terms of total amount. And confirming what we saw with the Wiesner test, when we look at these three metrics here, lignin to cellulose, S to G, and G aldehyde to G alcohol ratio, we see that both lactase 4 and lactase 17 are able to sort of compensate a substantial amount to total lignin deposition. Not quite to wild type levels, but close. They do restore S to G ratio, but only lactase 17 was able to restore the G aldehyde to G alcohol ratio, while lactase 4 was unable to do so, remaining sort of roughly at the level of the quintuple mutant even. Suggesting then that really this sort of bias in substrate affinity of our different lactase paralogs determines lignin composition, in this case specifically the ratio of the aldehyde to G alcohol units. And at this point, I also just want to mention work from the Dixon lab, which sort of came out in the same year as our preprint came out, which found a specific lactase here, like 8, which was responsible for the polymerization of that C lignin that I mentioned, that very specific seed coat lignin. And this lactase really was sort of much more active towards these C units than all the others, and in turn could not polymerize classic G units. So this was just sort of another angle that at the same time really made the same point as we did, as it so often happens. Now, if plants go through all this quote unquote trouble to really tightly control the composition and the amount of lignin in different cell wall layers and different cell types, that of course strongly suggests there's some sort of physiological relevance to that, right? That if you look at the fact that S units were evolved 4 times independently, that doesn't just happen by chance. At least the likelihood of that happening by chance is rather limited. So we needed, or we wanted to look at what the functional reason there was. Which of course brings up the first question, which is what is the function of lignin? There is of course many answers to this. One maybe more generalistic point is that lignin of course is required for the biomechanical stability of the plant, resisting lodging phenotypes and so on, and is associated generally with stem height, right? If you have a hyperlignified plant, they can't just grow very tall, they fall over. However, if you look at these sort of throughout published data from the last 20 years, if you look at the association between total lignin and stem height in both populus species and Arabidopsis, you see that while there is an association, it is very weak, suggesting that the total amount of lignin really is a rather poor predictor of stem growth, specifically in these lignin mutants. So that suggested that there was more to this association, and that actually lignin composition played a bigger role there than we perhaps thought. But to sort of decipher that, we thought that we needed to go into sort of more detail, into a higher resolution, both chemically and spatially speaking. So what we did is we looked at individual cells from both Arabidopsis and Poplar in the metaxylem, the secondary metaxylem, and the protoxylem. And we sort of characterized the shape descriptors of these individual cells. And then here, for example, you see these perimeters of different trackery elements from our different mutants, and you see that some, like the CCR1 mutant, as expected, many of them are terribly collapsed, while other mutants seem to be less affected. And then we took those same sections from those same plants, and in the same cells, we measured lignin composition using both Raman microspectroscopy and a Wiesner test. And then we took that data together with data about the tissue structure and the cell adjacency and anything else we could get our hands on, and try to sort of build models about which parts of lignin chemistry really affect the sort of structural integrity of trackery elements. Because of course, this collapse that we measured here, this sort of malformation, is induced by the negative pressures that are associated with the sap transport through the trackery elements, which then sort of pull on the sides on the walls of the trackery elements, and if they're not strong enough, they collapse inwards. building these models, we found, this is of course a simplification, but I don't want to bore you forever. We found some sort of recurring things that held true in both species and in most cell types and in most scenarios, which was that as expected, total lignin and G alcohol content strengthened the cell wall, while on the other hand, S units, which in angiosperms generally are excluded from TRK elements in the wild type, but they are accumulated in some mutants, S units weakened the TRK elements, as did the incorporation of atypical monomers. And also an extended degree of vessel adjacency. So if a vessel was close to many other vessels, that compromised its stability as well. One interesting thing here was the G aldehyde units, which sort of seemed to be very dependent, or the effect of them seemed to be very dependent on developmental state and cell type, having negative and positive effects in various scenarios. So we wanted to dig deeper into that a little bit, and did 3-point bending with mutants that were had an increased amount of G alcohol versus mutants that had an increased amount of G aldehyde residues in their lignin. And if we took the stems of these mutants and sort of did a 3-point bending test here, we saw that the increase in G alcohols increased the stiffness of the stem with no real influence on the flexibility. So the amount a stem can bend before it sort of snaps, while an increased amount of G aldehydes, as expected, dramatically decreased the stiffness of the stem, but also led to a dramatic increase in flexibility. Now, we also tried to confirm this with various other ways. One way was just molecular dynamics modeling. And here, at least the stiffness part, we could confirm, which was that these short artificial polymers of G alcohol had a much higher modulus, were much stiffer than the G aldehyde polymers. Now, the idea that a plant wants to be flexible is relatively easy to conceptualize, right? Because you don't want to just snap immediately when there's a bit of a wind blowing. But the question is, why would on the cellular scale, why does a TRK element need to be flexible? And this brings up an idea that originates from the Holbrook lab in Harvard, which is this idea that under drought conditions, the negative pressure in the sap transporting TRK elements increases, leading to the collapse of these TRK elements, right, as we've seen before. However, they have observed that under in certain scenarios, after the drought period, once water supply is restored, this collapse can be reversible. So these cells can sort of, you can call it reinflate, if you will, and restore themselves to the circularity of sort of pre-drought conditions. Now, of course, if you want to make a collapse reversible, you need to make sure that these TRK elements deform without snapping, right? They need to deform without losing all their structural integrity, still being able to return to their original shape. Suggesting then that this flexibility might exactly be important for that, so that only with sufficient levels of G aldehydes, you have a cell wall that is flexible enough to allow reversible collapse. And if we looked at drought conditions of these different mutants, we saw that indeed the mutant with high levels of G aldehyde restored its circularity almost to sort of wild type levels after recovery, while the mutants with the stiffer but less flexible G alcohol enriched cell walls could not recover to the same extent. And that also was reflected in the sort of overall growth of the plants, where most of these G alcohol enriched plants actually died after the drought treatment, while all of these G aldehyde enriched mutants survived. Now that, of course, is just sort of a glimpse into the world of the lignin structure-function relationships. There's so much more work to be done here, but it gives us a first idea of one scenario in which the specific lignification directed by specific lacase paralogs has sort of a determinate effect on the efficiency of drought resistance, TE collapse and the reversibility of it in Arabidopsis in this case, meaning that this G aldehyde mediated flexibility really has a strong physiological importance for the plant. Which brings me to the end. So if I just briefly summarize what I talked about, is that the subcellular localization of specific lacase paralogs delineates lignification in different cell types. That these different lacase paralogs do have substrate biases or preferences, which in turn directs the lignin composition of the resulting lignin. That these different lignin compositions are crucial to determine cell wall biomechanics, and that these different sort of degrees of or different balances of flexibility to stiffness are crucial to maintain growth in changing environments. With all this, I want to and need to thank Edouard Pesquet, who was my supervisor through all of this, as well as other lab members of the lab, as well as collaborators in Tokyo, Shinjaki Jitano Dagula specifically, and my co-supervisor, Vincent Billon, as well as Cheng Shu Li at Umeå University. And you, of course, for listening today. And I'm happy to answer any questions later.
So thank you, Dr. Blaschek, for this great, interesting, and quite unveiling lecture on how lignin is controlled in the plant. So if anybody has any question, please type those questions in the chat box, and we will go back to your question at the end of this webinar. Okay. So now it's my pleasure to introduce our second speaker of the day, Dr. Norman G. Lewis, who will present a lecture entitled Lignin in the Biosphere and Beyond. Well, we could refer Dr. Lewis to, you can start smiling, no, that's okay. We could refer Dr. Lewis to as the Captain Kirk of lignin science. You all know that Captain Kirk from the original late 1960s Star Trek series. Maybe the youngest one don't remember that series, but we as kids, we were watching that series. And you could even look at the photos of Captain Kirk from that old series, TV shows, and a photo of Norm. In some way, they even look alike a little bit. So that's really amazing. But why this reference to Star Trek and this connection with Norm? It's because Dr. Lewis was the first scientist to study plant growth and lignin biosynthesis in space. Yes, in space, notably on the International Space Station in collaboration with the NASA. To give you a few words on who is Dr. Lewis, is the Regents Professor and Arthur M. and Katie S. Todd Distinguished Professor at Washington State University, where he notably served as the director of the Institute of Biological Chemistry for about 25 years. He has received countless distinctions and awards. I will not give you the list here, but it's quite amazing. He has been the president of the Phytochemistry Society of North America for quite some years, and he was also one of the editor of the journal Phytochemistry for about 25 years also. Among many remarkable scientific achievements that Norm and his team did, they notably, as Leonard was mentioning, actually, his laboratory discovered the dirigen proteins, notably in the context of the stereochemical control of phenoxy radical coupling in plants. So obviously it has to do with lignification of the plant. So Dr. Lewis has published more than 270 scientific papers and patents. So his record is really, really remarkable. So with all due respect, Norm, Dr. Lewis, the floor is yours. And so please, to go back to that Star Trek, reference, please bring us where no man has gone before.
Well, years ago when I was in Montreal, we had a problem with the projector, and somebody, when I was speaking, said, "Beam me up, Scotty."
Yeah.
If you recall that particular
I do.
comment, yes. So let me begin by thanking the organizers for the opportunity to speak. I'm going to begin also with a historical context and bringing this into the relevance to lignification. And I'm coordinating this presentation technically with Laurence Davin, who's sitting beside me. And I'd like to thank Leonard Blaschek for, making the point that different lignin compositions impart different biophysical properties, because that's something that it needs to be reinforced at length. So lignins, there are numerous papers, even to the present day, that indicate that lignins can be three-dimensionally cross-linked. They can be highly branched or slightly branched. They can be linear polymer. There's no other natural polymer that I know that has ever been described in this way. So let's then switch attention to the evolutionary history of land plants from algal forerunners. This is a graph by James Doyle. And the key point here is that from algae, the plants that evolved first were the non-vascular plants, such as the liverworts, the hornworts, mosses. And then we move into the lignifying organisms, lignophytes, lycophytes, ferns and allies, gymnosperms, and of course angiosperms. But the key point was the ones like Merchantia polymorpha, the non-vascular plant systems, came on land first. And there's about 20,000 different species of these. Whereas in the vascular plants, and an example is a tree very close to where we are, 3,000-year-old western red cedar, there's about 391,000, of which the vast majority, about 369,000, are the flowering angiosperms. When we next go to look at the history of lignin occurrence reports, largely from the 1950s to the 1980s, the gymnosperms and angiosperms I have in black because that was correct. But in red were all these different reports of lignins and algae, non-vascular species, mosses, sphagnum, etc., fungi. And they were mainly incorrect due to the technical or technology limitations in those days. But even today, we still see some of these types of reports appearing in the literature. So taking that point into account, why don't algae and why don't non-vascular plants have lignin? Because they don't have this monolignal biochemical pathways. And, they're absent in algae and the non-vascular plants and fungi. This particular pathway just shows the different biochemical steps. There's a lot of mutations and downregulation, etc., that we've studied the plants, as we've heard earlier, but not today. But all of these, most of these studies earlier on largely avoided this question of phenolic coupling. So let me point out now, from a historical perspective, who I view to be the phenolic one-electron oxidative coupling pioneers. The first one I'll begin with is Holger Ertmann. He looked at the oxidative coupling of eugenol and isoeugenol. He was also the chairman of the Nobel Prize Committee in 1973. And he studied under Nobel Prize winner Sir Robert Robinson. The next person is Carl Freudenberg. And Freudenberg brought a great deal of understanding of coupling of monolignals to give rise to dimers like 8,5, 8,8, 804, etc. And he also studied under the Nobel Prize winner, Emil Fischer. Moving over to the person who coined the name lignans, which were normally dimeric phenylpropanoids at that point, Robert Downes Haworth coined that name. And one of his students was Peter Pausen. And Peter Pausen was my undergraduate advisor when I was in Glasgow. Their pedigree descended, I guess you would say, from Johannes Lysenus. And if we go down on the other right-hand side, we come to Sir Derek Barton. But sorry, before I do that, I should mention about Peter Pausen. Peter discovered ferrocene, which was a tremendous discovery, but he got the structure incorrect. He thought it was ionic, but it was shown to be the first organic metallic pi-pi sandwich compound. And just by having that one structure incorrect, he didn't get the Nobel Prize, which went to those who classified it as a sandwich compound. So it's something to be thinking about as you move forward in what you do scientifically. You need to get things absolutely correct. Coming to Sir Derek Barton, he studied phenolic coupling, molecules like cousinic acid formation. And just for those who are listening, before chemistry, when he was 17, 18, 19, he spent 2 years as a timber industry apprentice. And fortunately for us, he turned to coupling, phenolic coupling, and among many other tremendous contributions to organic chemistry, which resulted in the Nobel Prize. But the interesting thing is that all these phenolic coupling reactions all gave optically inactive or racemic products. And I'm also making appreciation to Sir Derek Barton. He was a great supporter of our work. So moving on from there, how did I begin getting into this area here? Well, in 1980, I was trying to decide what research I wanted to do. And on the right-hand side is a picture of the previous director, Arthur C. Nish, a very prominent lignin biochemistry. And as I sat in the library, this portrait of his was staring down at me as I was trying to figure out what I should work on. So I don't know if that was some of telepathy. I then moved to Paprika, the Pulp Paper Research Institute of Canada, worked on lignosulfonates to begin with David Goring, and worked with Alex Bialski at a company called Tembec. And we were able to take their plant system into utilizing about 1 million tons of lignosulfonates annually, which of course folks in Scandinavia have been doing for a very long period of time. But at that time, we began to initiate lignin and lignan biosynthetic studies. So here's where the enigmas were in the early 1980s. Lignans were largely or frequently optically active. Lignins were viewed to being racemic, and proanthocyanidins and elygiotannins like procyanidin B2 and telemigrandin also have specific chirality and optical activities. So the question was, how was this controlled? So we focused on the lignin enigma. Our next development, many years later, was in 1997 with the discovery of what we called divergent proteins. In the absence of the divergent proteins, we get non-specific coupling, giving rise to racemic products, as shown underneath here. But in the presence of the divergent protein, we get, in this case, ray-ray coupling to give rise to plus pinoresinol. And the important parts are generation of the radical, generation of a bis-quinone and then intramolecular cyclization. Now, getting back to what Leonard mentioned a few minutes ago, was a few years later, but 2005, we proposed that numerous plant phenol metabolic classes were likely divergent protein derived, including lignin. That was 2005. Let me just say that lignin workers and researchers were horrified. But moving on, we then were able to pull out other divergent proteins. The one that I've just described previously gives rise to plus pinoresinol, but another one here from Arabidopsis was able to control, rather than ray-ray coupling, psi-psi coupling to give rise to the opposite antipode. Again, with the same processes, free radical formation, bis-quinone and intramolecular cyclization. So then we looked at the sequences of the plus pinoresinol and the minus pinoresinol forming proteins. That's shown here for the plus and over here for the minus. And we looked at these sequences and we swapped out one of the domains of the plus pinoresinol forming divergent protein with a region from amino acids 90 to 138. And we were able to convert the plus pinoresinol forming divergent protein into a minus pinoresinol forming divergent protein. And that told us that we pretty much knew where the active site was in these particular classes of proteins. After a tremendous effort, we finally got our first divergent protein X-ray structure. We were unsuccessful in a variety of different ones we tried. We got it with one here from P, we call it DRR206. It's an 8-stranded beta-baryl monomer that goes into a trimer. And then our European colleagues, Gasper et al., published the AT-DIR6, the one that makes minus pinoresnol, about a year or so later. This now brings us to the dirigent protein superfamily and land plant evolution. If you look at the colors on the graph that's shown here, they're colored according to the types of plants that they belong to. So, for example, the liverworts and hornworts are shown here in sort of purplish colors. The bryophytes are sort of greenish. The lycophytes, etc. Anyway, you can see that there's this classification of different subfamilies from what we call DIR-A to DIR-I. And I should call out George Bowman's group at UBC. They were the first to begin setting up this sort of classification, but this is a much more advanced aspect. The key point here was, though, that both non-vascular and vascular plant species contain large number of dirigent protein subfamilies. And when we look at the plus and minus pinoresnol dirigent proteins I've discussed, these were in what we call the DIR-A1 and the DIR-A2 respectively. So then moving ahead, that was giving us some idea about how control of phenolic coupling was occurring. Would it have any relevance to lignification? So other, Lou et al., one of our collaborators, looked in cotton with hemigossypol, and they found something very analogous to that of our coniferal or pinoresnol formation. In other words, generation of free radicals, coupling, in this case, not intramolecular cyclization, but rearomatization to give rise to an optically active plus gossipol. And the corresponding one giving minus gossipol has been described as well. Well, then that is in the DIR-D subfamily. In the lower two cases of what we call PSPTS1 and PSPTS2, this takes us into a different set of phenolic compounds. And this began through conversations I had with the late Hans van Etten, and we finished this work up. Unfortunately, he succumbed to cancer. But this was in the generation of pterocarpins, such as machian, and also this isoflavine called DMDF. The key point here is there's not a oxidative free radical formation as in the other cases here, but we are able to, through the loss of water, generate quinone methide chemistries, which is very reminiscent of the bisquinone methides. But in this case, we get an intramolecular Michael addition, for example, to give rise to, or cyclization to give rise to the minus machian molecule. We're fortunate to be able to crystallize both of these as the PSPTS1 from P that makes a minus machian, it's a trimer, and the one that makes the corresponding isoflavine, PSPTS2, is a dimer of trimers. So here, the important thing is that we now have three families that are involved in entry points to different classes of molecules like lignans, aromatic diterpenoids, and into pterocarpins and isoflavines. So we did sequence alignments. The first two here are associated with the pterocarpin isoflavine. The next two down, DRR206 and AT-DIR6, are involved with pinoresnol formation, and the last one at the bottom is involved in the gossipol formation. So we searched to look for where there were conserved residues. Some of these, of course, could be controlling how the protein folds, but others should hopefully give us some insight into the biochemical transformation. It's important to say we knew what the chemical transformations occurring were, but what were the residues likely to be that were involved in this? And in this respect, we've been unable to yet generate any of our X-ray structures with either substrate or product in them. So, and I'll get back to how we got to this particular point here. our collaborators, John Cort, Pacific Northwest National Lab, did some docking of both coniferal alcohol radical here and the bisquinone methide. The aspartate D40 up on the right side here, down to the arginine 141, has got a span of about 12 to 14 angstroms apart, just enough suitable to bind a coniferal alcohol radical, which is about 8 to 10 angstroms. And the asparagine 134, I don't know if it's in this particular figure, but anyway, it was proposed to reprotonate one of the bisquinone methide carbonyl oxygens to facilitate the nucleophilic addition of the C9 hydroxyl group to the C7 in the quinone methide. And I'm not going to go into the details of this, but we've done docking of the substrates that form minus Machian. We think we can deduce likely what these conserved residues are doing in Machian formation, and also in the next case here for for the DMDIF, the isoflavine generation. So now I would like to switch focus here, and I'll just want to show this confocal microscopy work we did on lignification in loblolly pine. And the important thing here is at the very top here, you can see the cambial region, and then as you go further down, you get into more heavier lignified levels of lignification. And what occurs here, and this comes back to what Leonard was saying, in this case here, initiation occurs at the furthest point from the cambium cells, and it then begins to move towards, both ends here begin to move towards each other, and then it begins to be initiated here in the middle of this hexagon that's shown here, and then it continues and continues in a very programmed way until lignification is completely completed. And this, I got to emphasize, was very, very different than originally envisaged. And other people like Alan Wardrop and Lloyd Davidson have noted similar sorts of things as well, bringing back or emphasizing the point that Leonard was making earlier on. So now I want to turn to what I will call the great results that were by David Salt and Gao et al. That was published in 2023 in December, I think it was, looking at Casparian band DIR-E subfamily lignin disruption. So in the wild type, or the call O0, if you look here, there's uniform lignification that occurs there. I'm not going to go into all the different mutants they have. I'll just come to the DIR sextuple mutant. And here you can see it's what I call punctuated lignification. Lignification is no longer able to continue. It's perhaps beginning at what I call these lignin initiation sites, but then it terminates. So this anyway helped confirm our hypothesis that dirigent proteins were involved in lignification. But the question now is, but how? You know, we don't know exactly what they are doing in the DIR-E subfamily, and with what substrates are being utilized. When we compare the DIR-E sequences, which is these DIR 16, 18, DIR 10, DIR 25, 9, and 24, we can see very similar residues that are being conserved, but there's one major difference here. The arginine that we see for pine-resinol formation and for pterocarpin formation is replaced with a serine, and we will need to establish how and what effect or what the role of that serine is relative to the arginine. So if you look at the different dirigent proteins in the A, B, D, and lignin forming DOE, one really important part here is the size of this beta-1, beta-2 loop. It is very, very large, and we are really interested in what that has to do in the DIR-E subfamily. Is it interacting with substrate? Is it acting as a plug? Is it what role does that loop have in addition to what we already know about the active site? So there are many, many questions that we still need to get straightened out with respect to the chemistry or the biochemistry of these particular processes. So I'm going to switch topics here again quite quickly. I'm going to go to lignin in space. And the question we asked ourselves was, is lignin needed when we go into space, whether for colonization or long distance deep space missions? So we generated a whole series of aragonate dehydratase mutants, and you can see on we can reduce these down to about 70% reduction with our quadruple mutant, which we call ADT 3456 here. And you can see that going from wild type, which can stand up erect, down to the ADT 3456, this plant is prostrate. So the question we had was, well, what would happen in microgravity? We have what would happen in microgravity? This is us growing the plants on the Advanced Plant Habitat on Space Station. We grew them for a 6-week period. We had 2 grow outs, and we could follow how these plants were growing, how the stems were developing on a day-by-day basis. This was part of a multi-omics process, which I'm not going to talk about, but the question was, how will they function in microgravity? There's no need to support, stem weight. Well, what occurred was that the plants with low lignin contents, beginning with the quadruple mutant, were unable to, they attempted to stand upright, but couldn't do it. And the less lignin there was, the quicker the stems went up and then basically went back onto the surface of the where the root tissues are. And this occurred in both systems here. So this recognized to us that there has to be a threshold, a lignin threshold, even in a microgravity or space environment. So the lowest lignin mutants couldn't stand upright. When we looked at the wild type, Kennedy Space Center, these cross sections are not great because they were frozen and they weren't prepared that you would normally do for this kind of microscopy. But you can see that the vascular bundles are all looking intact and normal, and similarly for the wild type on Space Station. Similarly for a quadruple mutant, the vasculature looks normal. But in the plants that couldn't stand upright, you see this asymmetry in the vascular bundles. So in other words, the plants were reinforcing one side, like you see perhaps in compression wood or tension wood, to attempt to stand up straight, but they were unable to do it. So the vasculature was asymmetric. They tried but failed. So lignin is required to some extent in space. This brings us back to the evolutionary history now of land plants from algal forerunners. First point I want to remember is non-vascular species evolved first. Second one is non-vascular species contain many examples of divergent proteins. They have specific subfamilies, and non-vascular divergent proteins are poorly understood, as are on the emphasis, the non-lignin cell wall phenolics. So going to this, you may have seen the nice paper from Keith Davis and Kathy Schwinn, etc., on looking at divergent proteins in the liverwort, Marchantia, and they speculate that the polymer that's in the cell walls in this liverwort are urinidin, and they suspect that this is a divergent protein control process too. And on the right-hand side here, you can see the different subfamilies and examples here of the divergent proteins that are present there. So the question, of course, is, some are in DIR-F, some are outliers, some exist between DIR-E and DIR-H in our way we've generated these graphs, but what do they all do? So let me just say, our belief in dirgent proteins is that we wouldn't be here as such. Land plants wouldn't be here as such if it weren't for dirgent proteins. So these are exciting times. Some of our current dirgent protein studies just now, we have several hundred constructs that JGI generated for us. So if anybody's got any interest in dirgent proteins, they should contact us. We might be able to help them on this. We know that monocots are specific, their subfamilies. We're doing some collaborative work with John Cotwell, Chrysler, and James Fulcher to try and understand these. The Duri biochemical characterization, we're doing grape and Arabidopsis with Yikong Rao and Jin Quang Huang. They were going to come over and work in our lab, but the US Embassy would refuse them any visas to come into the country. Liverwort DRF and other subfamily members, biochemical characterization. We began doing this with Kevin Davis and Kathy Schwinn. We'll have some work on that we'll discuss later. And we're hoping to get better dirgent protein structure determinations, perhaps with substrates, products, inhibitors, etc., with folks at Redwire here and Clyde Smith, who's done a lot of our has done the crystal structure determinations at Stanford. And we have an experiment coming on with low lignin alfalfa nitrogen fixation, because we believe that nitrogen fixation and generating legumes with low lignin might be the thing to consider in deep space missions and colonization of Moon or Mars, where fresh food is going to be there. So going to the next slide, it's acknowledgements and thanks. Without these particular agencies, we wouldn't be able to do what we are able to do. Began with the National Research Council of Canada and these different entities. So just going back sorry, keep going. So acknowledgements. Hans van Etten got us into the pterocarpin work. Diana and his student Rhodesia Seloy, Diana Bedgar, Mike Costa, Ki-Won Kim managed to get the first crystal structure for us. Chin-Yang Meng has done many of the others with us. Saeed Moinuddin is our chemist. And I also want to call out John Cort and Clyde Smith. They've both been really helpful in modeling of the dirgent protein active sites, etc. Clyde Smith getting the running and getting the structures for us. Christophe Hanot in France got involved in some other aspects here. And we have a new student here, Sophia Matilis, will be doing some work with us. And Howard Levine at NASA Kennedy Space Center is also been very supportive of our work. And one of the other persons that, in addition to ourselves, that had consideration that lignins were under control formation was Seymour Sarkinen, who unfortunately passed away some years ago. So on that, and Mike Costa, I may have forgotten. On that, I really appreciate the opportunity to discuss what we've been doing, and thank you very much.
Thank you very much, Dr. Lewis, for this great and so interesting talk about lignin in all kinds of situations. So I'm sure all we have questions from the chat boxes, and so maybe we can start with that.
Okay. We have a few questions. Let me ask the first one to Leonard. Could you please explain how to measure stem elasticity prepared with aldehydes enriched lignin?
Yeah, I mean, I guess there's two ways. The technical, I mean, the 3-point bending essentially is just you end up with a sort of load displacement curve, right, where you see sort of how much weight is on the stem, and then you see how much the stem actually gets displaced, and that curve has sort of a very very smooth shape until at some point it breaks irreversibly, right? And essentially that point of breaking, the displacement that the stem can undergo to the point of breaking is what we call flexibility, right? How much can it bend without irreversibly breaking? The other half of that answer would be, how do you get the aldehydes in there, essentially? And that's just, or at least in our case, we just use mutants for that, right? So we have different mutants, different monolignal biosynthesis mutants, which are specifically reduced or enriched in C aldehydes or C alcohols. And those we could sort of compare with each other and get an idea of what these monomers did to the biomechanical parameters of these plants.
Great, thank you very much. And before I continue on the question, I want to thank you both for your great presentation. It was really clear, really nice to see and to learn about lignins. So we have another question for Norman. Do dirigent protein always form homotrimers, or can they form heterotrimers to alter the stereospecificity of the phenolic assembly? As in Arabidopsis, deer form a multigenic family of more than 20 paralogs.
Yes, let me try to answer that question. Certainly in the cases of the pineresinol forming proteins, it stops at the dimer, yes? But the DIR-E molecules that are involved in lignification, we don't yet know. We have no evidence in vitro biochemically as to what substrates they utilize and what products they generate. I think it's safe to say that in cases like in the liverworts, where you've got molecules that are derived from orunidin, that process does lead to what in our preliminary work here, or work that we're ongoing, seems to generate some sort of polymeric entity, but the extent of which we've not yet done, you know, based on molecular weights, etc. So I think the key point here is what there's a variety of different things that one has to consider. One is, how are the different molecules of different molecular weight being generated? Is it reoxidation, for example, if we're growing lignin chain, or if we're growing oligolignan? Is there other processes going in there of you know, like a chain reaction? Are there processes like electron tunneling occurring? These are the questions that really need to be understood. The point of the Casparian band lignification, with the interruption of the generation of the continuous Casparian band lignin deposition there, hopefully provides some promise to be able to answer that question. Does that make any sense?
I think so. Thank you. Stéphane, do you see a question for Norman?
No, but I can ask some question also, or?
Yeah.
Yeah. No.
Yeah. In the chat box, you should also be able to see some questions.
Oh, okay. Okay. To
But go
Norman
ahead.
or to Leonard?
To Norman.
Okay. Yeah, no, I don't have anything additional from you. Oh, yes, maybe. No, yeah. Okay. There is a question from them. Given that there is no falling down at zero gravity, do you think the observed phenotype could be negative phototropism, perhaps induced by cell wall integrity sensing modules? Oh my God.
Well, I think we have to consider that lignin does more than structural support. You know, it helps waterproof piping so that water nutrients go from the roots up to the, you know, the treetops, depending on the organism. So whether reducing lignin makes the water conducting vessels, for example, leaky, you know, like a straw with holes in it, and what effect that would have, but there's something most definitely occurring and in microgravity. And remember, these are in tightly controlled advanced plant habitats with lighting, etc. They don't function that way with the wild type, and they don't function that way with the ground control. So it appears to be what we see. The plants just don't have the strength to be able to stand upright with very, very low lignin contents. And I'll make a point here before I forget. I mentioned that Robert Downes Haworth coined the term lignin. I forgot to say this. And then I worked as an undergraduate for Peter L. Powson. Well, as we move down the pedigree line, here's the book on lignans that we have here. It's called the Lignin Handbook. Took us an enormous amount of time, but it's a great testimony to work that Robert Downes Haworth began in the early 20th century. So that's a point I'd forgotten to mention.
Okay.
Thank you, Norman. I have a question that's to both of you, I think. So first, thanks for the wonderful talk. My coworkers at the University of Florida are watching your talk together. We are working on food chemistry here. I know that high lignin content can make food taste less pleasant, and that reducing lignin content might make edible plants more susceptible to stresses. My question is, what are the significances of lignin study in food sciences, and what are the possible future directions?
Yeah, I could begin to address that point. The reason we would like to be able to grow plants in space, for example, with low lignin content, is so that we could be able to generate plants where the edible parts could be eaten and inedible parts could be more readily recycled. Also to look at nitrogen fixing plants, because they would provide an opportunity to not have to bring up huge amounts of solid or liquid nitrogen containing fertilizer. The question as to the taste of lignin, obviously anything that's high in lignin content is more woody, and really for us it's indigestible. So it's a question of without knowing specifically which plant species they were thinking of in food, but even for things like alfalfa, to be able to reduce the lignin content there has been shown to result in better digestibility, etc. So there are certainly advantages to lower lignin contents in the cattle and livestock industries. Now, with respect to stresses, this is another question that we have, and I'll say both down here on the ground and up in space. We can provide and generate these low lignin lines, to be grown up in space, but what effect does cosmic radiation have on the plant? Because phenolics tend to protect against this, or do we need to have a combination of less lignin, let's say more soluble phenolics to offset that? And similarly, with low lignin plants that have been generated as mutants, etc., they normally show some sort of they're not wild type. There are very few of them that have turned into commercial products. So that would be my first thoughts on that we need to consider whether stress responses are which you might be able to detect at the transcript level, but is it really being manifested through at the protein and enzyme level and then to the metabolite level. And we know from experiments we've done with knocking out these orogenate dehydratase enzymes, the phenylpropanoid pathway is upregulated. So it's showing a very strong transcriptional response, but the plant is unable to make the monolignol. So it's a futile response, if I'm making any sense. I don't know if that's a partial answer, and Leonard, I'm sure, can now give you the real answer. Yeah.
No, I think that, I mean, if I had to add one thing, I think is that where we go from here, I think we'll have to involve much more specific approaches. So often in the past, whenever you went on, modifying lignin in plants for any goal really involving food science, you would sort of take the sledgehammer and just sort of knock out lignin monomer biosynthesis or something, which of course has all the effects that Norman just outlined. But if we get a little bit more sophisticated about what we're planning and sort of try and find ways to actually modify lignin monomer biosynthesis and or polymerization in specific tissues, specific cell types, and at sort of specific developmental sort of time points, then I think there is definitely potential there to find something that has minimal effect on the resilience of the plants while still improving whatever parameter we want to improve for plant usage.
Right. Thank you very much. We are running out of time, but go ahead, Stéphane.
Yeah, I have a question for Leonard. Leonard, what about the peroxidases?
I mean, that's good to ask that question with less than -1 minute remaining. If you have another half an hour. No, quickly speaking, I think the fact that the quintuple mutant that we showed was so drastically reduced in lignification leaves me with two, again, very briefly put hypotheses, which is one, the peroxidases are really mainly just important for the tractary elements and not the fibers. But what I think is more likely is that what the peroxidases do in lignification is essentially directly dependent on lactase activity. So they might act in some sense downstream of lactase activity in the sort of timeline, in this very specific timeline of lignin polymerization that Norman outlined so nicely. But what exactly is going on, I can't tell. Those are my hunches.
Okay, just to go on with the lactase now. I mean, so I understood that there are different lactase with different specificity, capable of making different type of lignins. But you also have two types of monomer in terms of the function: the alcohol and the aldehyde. Okay, and if you have the aldehyde, the inherent chemical reactivity of those aldehydes are different from the alcohol. The aldehyde is conjugated, up down to the phenyl ring thanks to the double bond. So the redox properties of those monomer will be drastically different from the alcohol. Have you seen any correlation between the capacity of such lactase to oxidize or not, an alcohol versus an aldehyde? An aldehyde should be, yeah.
Yes. So I mean, some of that work, well, not with lactase, but with how these once oxidized will couple has been done, of course, by Ralph and colleagues. But one of the reasons that we really haven't gone into that is that and there might be people disagreeing with this, but in my mind, we still don't know what the lignin monomer actually is. And by that, I mean the functional lignin monomer. We don't know what really is oxidized by these lactases and in what form and in what shape. Just because when we try to make lignins in vitro with sort of lactases, I mean, for example, I showed you the work from the Dixon lab there on the cafeol specific lactases. The lactases that supposedly are responsible for the formation of G-lignin that they identified there and that they sort of purified, they could only oxidize the supplied G alcohol in the presence of the watery extract of the whole tissue, right? Without it, they couldn't, which really just tells you that there is more going on. It's not just one lactase and one substrate. It is happening in a context that we still don't fully understand, and that might involve mediators, right? So we might have scenarios where lactases oxidize small molecules, which then transfer that radical charge to the actual substrates. Yeah. But this is such an enormous question. which we sort of knew how big it was going in, so we focused on the effects in the plant, if that makes sense. What actually the chemistry is of these scenarios, we still don't fully understand. And
and you might also have divergent proteins.
Yes, exactly, of course, and I mean you probably do. But how all of that in the end plays together, I'm very curious to see.
It's very complex picture. Yeah.
All right, we are out of time, so I'm
Can
sorry.
I ask one last question to Norm?
No, I think it's too late. But if you have a question for Norman or for Leonard, everybody feel free to email them directly or email us, and we will share that question. So I would like to thank all our fantastic speakers today, Dr. Blaschek and Dr. Lewis, for your great presentation. It was awesome. But also Dr. Quideau for moderating that session. Also, I would like to thank Dr. Salminen, who is managing the recording and advertising the webinars, and also organizing the ICP conference in Turku in July. Also, I would like to thank Dr. Baron, who is our president of the Groupe Polyphenols, who was at the origin of the webinar initiative. Thank you, everyone very much for your participation, and we hope this webinar was helpful and it brought some new knowledge to you. So please send us an email, as I said, if you have any questions. The presentation are going to be available free of charge on the Groupe Polyphenols website, on YouTube, on Instagram of the Groupe Polyphenols. Before closing the webinar, I would like to remind you to please just take 2 to 5 minutes to complete the survey. You're going to receive a link, an email in a few minutes, I think, to access that survey. Otherwise, you can scan the QR code that you have on the screen. And I would say thank you again, and stay tuned for future webinars. And we look forward to seeing you at ICP in Turku, Finland in July. Thank you, everybody. Have a good day.
Thank you. Bye-bye, everybody.
Bye.
Bye, everyone.